Cells are the fundamental units of life. This potato consists of plant cells.
This pond water is filled with single-celled organisms.
But to be able to understand cells, we have to see them.
The development of the microscope has advanced our understanding of cells and of life immeasurably. A lot of medical research takes place at the cellular level.
Hi, Ellen.
Hi, Julian.
Could you tell me a little bit about the research that you do?
One aspect of the research that we're interested in is looking at how cells divide normally. And we use a number of different experimental research strategies to look at how cells divide. And we hope that what we learn from normal cell division will have implications when we look at cells that are abnormally dividing, such as cancer cells.
What type of microscopes do you use in this type of work?
Well, we use three different types of microscopy. It's a small portion of the type of research that we do here, but it's one way in which we analyze samples to determine at what phase they are in the cell cycle. We use light microscopy, we use fluorescence microscopy, and we use phase contrast microscopy.
So to look at potato cells, I'll need a light microscope.
And a wet mount slide of a specimen.
Right. Here's a glass slide, a cover slip, a dissecting needle, and an eyedropper with some water. Now you can prepare a wet mount slide.
First, I'll cut a small wedge potato with a scalpel.
Then smear some potato cells from the wedge onto the center of the slide.
Add a drop of water.
Touch the cover slip to the water and lower it onto the slide with the dissecting needle.
Okay.
What do you see?
Just a smear. I don't see anything distinctly.
Oh, the cells are there all right. They just need highlighting. You have to add a stain.
Fluorescence microscopy is based upon the principle that you use special dyes that will bind to particular structures in the cell, and then you can identify those structures by virtue of exciting that molecule by exposing it to a particular wavelength of light. And then once that molecule has been stimulated, it will give off a different wavelength of light, which is then visible. In our lab, one of the stains that we use is a fluorescence dye that specifically binds to DNA, or the genetic information within the nucleus of the cell. And in that way, we can specifically look at the changes that occur in the DNA throughout the different cell cycle phases.
A wood stain brings out the grain in this cabinet, and this dilute iodine will highlight cells of the potato.
So instead of water, I'll add a drop of iodine. I see something.
So the grains of starch in potato cells are highlighted by iodine. Some stains are used for plant cells, but others work better with animal cells, such as this nerve cell.
The nucleus really stands out.
In our work, we use phase contrast microscopy to look at other facets of the nucleus as it's going through different cell cycle phases. We've already used a fluorescence dye to identify and visualize the DNA in the nucleus. We use phase contrast microscopy to visualize the nuclear envelope, which is an envelope, a membrane that surrounds and contains the DNA, which is a very specific structure which only occurs during a certain stage of the cell cycle, and it's very important for structure to be formed properly in order for normal cell division to occur.